Good morning, friends, and welcome to today's edition of Anti-Doping Course brought to you by NPTEL in association with Madras IIT. So, this is a continuation of week 7 lecture number 3. And today we are going to discuss on genetic doping control. I'm Professor Dobson Dominic, a sports medicine specialist. The previous lectures in week 7, we looked into what are the strategies we can adopt for a proper anti-doping control. We looked into the main strategy adopted by World Anti-Doping Agency, that is the athlete biological passport. And in this lecture, we are going to look into how we can minimize the newer forms of doping strategies adopted by athletes. That is, the latest one is the genetic doping. We'll look into that in this lecture. So these are the broad learning outcomes in today's lecture. What are the doping relevant genes? Very important to know what are the genes which the athletes misuse as a form of performance enhancing genes. So doping in this new modern era has moved from performance enhancing substances or performance enhancing drugs or even performance enhancing methods to the newer latest performance enhancing genes, that is gene doping. We'll see what are the different mechanisms by which this strategy is adopted, that is the gene doping mechanisms. We'll look into what are the difficulties the anti-doping agencies have in detecting this newer form of doping, and we'll conclude with an update on the future of doping control, the way ahead. So to begin with, we'll look into what is a sports gene. Genetics, as we know, has a very large influence on athletic excellence such as strength, muscle size, muscle fiber composition, whether it's a slow twitch fibers or a fast twitch fibers, anaerobic threshold, lung capacity and flexibility. Now, if you see the effect of genes on the structure, function and performance, certain morphological characters or kinetochronometry like height, length of arms, the effect of genes is very large. Okay, so this becomes beneficial for certain sports like basketball, swimming, certain things, morphology like lung size and volume, again dependent on genetics. The implications of movement speed, balance, flexibility of joints, all the genetics has a very important role. Even in lung size and volume, the accuracy of movements, the reaction time, the flexibility of joints. So pretty much all the performance characteristics for an athletics, genes play a vital role, and hence the athletes misuse genetic doping in the expectation to improve the performance. So what are the doping relevant genes which athletes have been trying to influence? The most important ones are the speed genes, ACE gene, ACE gene and ACTN3 gene, which are coding for the fast twitch fibers. Okay, especially the ACE D gene is involved in fast twitch fibers, and similarly ACTN3 gene, which are predominantly seen in certain Caribbean athletes like Jamaicans, you know, Usain Bolt, certain countries like Trinidad and Tobago and Jamaica. There the athletes are blessed with a lot of fast twitch fibers. Again, it's got due to the genetic influence, especially ACE-D gene and ACTN3 gene. Similarly, certain areas like Kenya, Ethiopia, in Central Africa, there the athletes are born more with some slow twitch fibers gene like PPAR delta. Okay, these are all present in skeletal muscles and adipocytes, they promote fat metabolism and increase the number of slow twitch fibers. Very important for endurance events like marathon running. Even for strength-based sport like weightlifting and powerlifting, certain genes becomes very important like myostatin. Okay, so this gene regulates skeletal muscle and inhibition of muscle size, power and recovery. Okay, so in these genes, if it is coded well, then the athletes are beneficial. So we looked into the different genes which influence the athletic performance. Now, how do gene doping happens? Gene doping refers to the use of gene therapy. There is something called gene therapy, which is a medical thing, okay, which is used for certain medical conditions like congenital conditions. Okay, so this gene therapy is misused by athletes in the prospect to enhance the athletic performance. This involves gene doping involves the modification or introduction of genes into an athlete's body to increase the production of specific proteins or hormones which can enhance the muscle growth, muscle size can increase, muscle endurance can increase, or even they recover fast from a certain bouts of exercise. Some form of gene doping helps in recovery, okay, so that enables the athlete to train multiple sessions. So we all know the importance of recovery for a professional athlete. Gene doping has a potential to improve the recovery performance of an athlete. So gene doping has a possibility to improve the muscles growth, endurance, muscle endurance, and also a recovery of an athlete. So if you look at the history of gene doping in sport, gene doping initially came into the picture from gene therapy. And in 2001, first official discussion by the gene therapy working group under the International Olympic Committee was started. In 2003, gene doping has been explicitly prohibited under the regulation of WADA. In 2004, WADA established a committee to investigate the latest advances in the field of gene therapy because they understood the implications of gene doping in the performance, and they suspected already that widespread gene doping was happening in the sporting world, and WADA constitute a committee to investigate and do research in the latest advances in the field of gene therapy and methods of detecting gene doping. So WADA also published a laboratory guideline in 2021 for gene doping detection based on PCR or polymerase chain reaction, indicating the establishment of standardizing method on the way. Okay, so thereby WADA has recognized the importance of newer technologies to identify gene doping at the earliest stage. So this is the history of gene doping. If you look into the mechanism of how gene doping happens, the one important one is by gene modification. It involves altering genes to improve the body's physiological response to exercise or to increase the efficiency of energy use. There are several techniques which may include transfer of genes that promote muscle hypertrophy or muscle growth, or enhance oxygen delivery through increased red blood corpuscle production. So both in terms of resistance trained athlete, also there is implications for gene doping. Similarly, for aerobic athletes like cyclists, marathoners, or skiing, also there is a implication through gene doping. Potential methods of gene transfer, gene doping. One is direct gene transfer, whereby introducing desired genes into athletes' cells using a vector or a medium such as virus. The commonest virus or the commonest vector are adenovirus, which is the most common one. Then retrovirus is there, lentivirus is there, adeno-associated virus, whereby these is inserted directly into the genes using this vector. The second method is through cell therapy, where the modified athlete cell in a laboratory and then reintroducing them into the body. So it can be in vivo or ex vivo. So these are the two techniques by which gene doping methodology happens. Types of mechanisms of gene doping. One is through somatic gene editing, whereby making changes to non-reproductive cells, that is the somatic cells. Genetic modifications are not passed on to the future generation. So this is somatic gene editing. The second one is the germline gene editing, whereby making changes to reproductive cells. Here the genetic modifications are passed on to the future generations. The types of gene doping: gene expression modification, where there is a modifying expression of existing genes rather than introducing new genes through several techniques like RNA interference, RNAi, or the latest CRISPR-Cas9 gene editing. The final one is through gene transfer, where there is a transferring of genetic materials like DNA or RNA into an individual cell to enhance athletic performance. All these forms of gene doping constitute a sports doomsday scenario, a new generation of bioengineered performance. Imagine athletes injecting artificial genes right into the muscle. Scary. So it literally becomes a doomsday scenario. Now, there are risks in gene doping, starting from ethical concern. So WADA has a principle of clean sport. So there should be fairness in competition, and with the advent of gene doping, it is threatened big time. And there is also access and inequality. So if there is some countries adopt new doping strategies like gene doping, then obviously they are at an advantage and there is inequality. Gene doping also has several health risks for the particular athlete. There can be possibility of unintended genetic mutation which could lead to health complications, including increased risk of carcinoma, cancer, and also immune disorders. So there are several health risks which are involved in gene doping. Now coming to detection techniques, how is gene doping detected? So this is through something called genetic and epigenetic analysis. There are several methods which the labs try to identify gene doping are either it's through direct method or through indirect method. The direct method, the most common is PCR, which is polymerase chain reaction Southern blotting technique, whereby the detection of the inserted vector or by detecting the inserted gene's length. Okay, so this is done through PCR technique, and indirect detection of gene doping happens by identifying quantitative transcriptomics or through identifying quantitative proteomics. Also, this is through different techniques like real-time polymerase chain reaction or through mass spectroscopy and 2D electrophoresis, which are all latest gene doping detection techniques. It can also be done through an indirect method where we observe the immune response through techniques like ELISA and Western blot. What are the challenges faced in detecting gene doping. Detection methods for gene doping are still under development, making it difficult to identify athletes using these advanced techniques. The biological passport monitoring, although it helps in detecting athletes' biological markers, identifying specific gene modification remains complex. Okay, so the athlete biological passport is more for looking at the athlete's biological markers like urine changes or blood changes, but identifying specific gene modifications remains complex. So these are the references for today's lecture. Thank you and Jai Hind.